So cephalochordates are a phylum, right, within the supraphylum Chordata. And so we did phylum Urochordata. We're doing this phylum now, Cephalochordata. And this is what a cephalochordate looks like. This is a live one in an aquarium. It's has a name called Amphioxus, is another name for it, which means pointed on both ends. Amphi means pointed, oxus means both ends. So they got a kind of a pointed cranial end and a pointed tail. You can tell the head because the head has some specialized structures up here for feeding. And it has a little like knob here on the head. This is called the rostrum, which means kind of nose. Rostrum means nose. And what's cool about this shot is this little structure that's sticking out and supporting the inside of the rostrum is the top of the notochord, right? So, or the cranial end of the notochord. So it goes all the way into the rostrum, giving support to that. And it will continue all the way along the length of the amphioxus here. Now, on top of that is the nerve cord. And you can always find the nerve cord in these guys because of these darker cells. They form the dorsal surface or the back surface of the dorsal hollow nerve cord. And then the rest of these kind of structures or stripes along this are myomusculature. We're going to go over all of these structures on the slides. And this rostrum continues as a dorsal fin, which goes all the way back to the tail that helps with locomotion. So we're going to talk about all of the specializations to these guys. So Cephalochordata, there are lots of fossils, wonderful fossils, particularly that kind of start to talk about or give you some information on the origin of chordates. And chordates themselves, they're an old lineage. So chordates arise in the fossil record about 540 million years ago, during something called the Cambrian explosion, right, where you get all these groups of organisms diversifying because of the development of structures like the notochord. And so this is a more derived species of cephalochordate than even Amphioxus, that is a fossil. Looks like a snail, really, right, with its specializations to the head. It has a pharynx and pharyngeal slits, and it shows all of the chordate characteristics as an adult. And its name is Pikakia, which is named for the area in which it was found. And this is kind of what the fossil looks like. And of course, they've done CT scanning on it and built some nice three-dimensional models of it. The living group of Cephalochordata is within this class, all right? So phylum Cephalochordata, class Leptocardii, all right? So Leptocardii means basically it's talking about the pierced pharynx here. And these guys are all marine. They're all shallow coastal water organisms. And so they will always tend to orient themselves like this. If you put them in an aquarium or if you're searching for them, they basically bury themselves in the sediment to hide from predators with their heads sticking out. All right, so the head sticking out is basically allows them to filter water and breathe, and also to find food. So they're going to be a lot like a Urochordata in the sense that they filter feeders. They're filter feeding organic material from the water column. But they tend to want to be buried like this. And we'll talk about a specialization to their notochord that allows them to do this efficiently. The other name they go for is Branchiostoma, all right, and this is the actual genus name. So branchio means gills, stoma means mouth, so gilled mouth. And so their mouths lead directly to their pharyngeal slits, which act as gills and a filtering mechanism. That's where that name comes from. So this one shows you a cartoon of them within the sediment, and then is going to name a bunch of structures that we're going to go over. What's nice is a lot of the names are the same as the Urochordates and have similar function. So So here are the drawings kind of out of the textbook that I suggested you guys might want to look at. But again, you don't really need to purchase it because I give you kind of all the structures as long as you're in lecture, right? Which you all are showing up, so that's good. So we got all the 5 chordate characters, and then we got a list of terms here. All right, some of these you've seen, some of them you have not. And so we got a sagittal or side view of these guys, and then we're going to look at a number of cross sections. And then in lab tomorrow, we'll do the same thing. We'll look at these guys under a microscope, because I got small juveniles and they've been stained, so that you can ID all these structures, or at least the ones listed on the lab handout. And we'll also look at cross sections through certain parts of their body. And I have a really good atlas online for that also that I made a while back that labels everything for you. So you'll see both of all these characteristics or anatomical structures in lecture and lab. The first term up here is pediogenesis or pediomorphic, is a term that means child birth, basically, when you look at the Greek, which means they are retaining larval characteristics as adults. And this differentiates them from urochordates. So remember, with the urochordates, you had an adult stage that was benthic, right? That was attached to the substrate. And then you had a larval stage that could swim. Well, these guys simply swim as larvae and swim as adults. They just retain the same characteristics that the larvae have and grow to adults, and then can reproduce. So that's what pediogenesis means. We'll see it again with salamanders. There are certain salamanders that stay in the larval stage their entire life cycle, which means they breed in the larval stage, right? And that's where that term comes from. Now, when you look at these guys, so this one is the that fossil, right? Picaia, right? And this is the living Amphioxus. And when we look at it, these are all of the structures, most of them listed over here. We have a pharynx, a really large pharynx that's closer to kind of the cranial end of Amphioxus. And it's pretty huge as far as the structure goes. And it, again, sits within another sac or chamber called the atrium, just like with the urochordates. And it has slits and pharyngeal bars, and they're going to be lined with cells that produce mucus, and those cells are going to be ciliated, and they're going to beat, right? And help move water into the mouth. So the mouth is underneath the rostrum here. There's a large round mouth, and then there's this oral cavity. Sometimes it's called the buccal cavity, if you remember that word from last class. So buccal means mouth or oral space. And it's often called the buccal hood or the oral hood. And within that large space, there is a specialized organ that's kind of unique to Amphioxus that's called the wheel organ. And it is heavily ciliated. And when this animal's alive, it looks like they're doing this. They're spinning or moving. And that's because the cilia are beating in unison. And so that's why they call it the wheel organ. And what that does is drive water or suctions water from the environment into the oral hood and then into the pharynx. Once in the pharynx, the water will slip through the pharyngeal slits into the atrium. And any food that comes in with this, that's suspended, right, organic material, will stick to the mucus that's lining the bars and then be collected and pushed to the intestines, which are back here or caudally located. So you can see the pharynx kind of empties into what they call an esophagus, and that esophagus then drains into an intestine and into a cecum. I'll show you some pictures of those in a second. So the way this works is there's an endostyle, just like we talked about with the urochordates, and the endostyle makes up the floor of the pharynx, and it's a groove. I'll show you a picture of it in a second. We know that it can produce thyroid hormone, concentrate iodine, but it also produces lots of mucus. And then that mucus is spread upwards and around the bars. On the top of the pharynx, or the dorsal end of the pharynx, there's going to be another groove that we'll talk about here in a second that collects the mucus and then pushes that mucus backwards towards the esophagus and the digestive tract. And so you got mucus being produced all the time, spread out, collected, and then pushed dorsally. And all the food that's stuck in that mucus then ends up in the intestines. All the water that passes through the pharynx goes through the slits into the atrium. And then the atrium drains through a pore back here called the atriopore. All right, so that's basically where all the water leaves. And then notice if you go further back, the intestine eventually empties through an anus. And then there is a post-anal tail, like we talked about, one of the chordae characters, right? That propels this organism forward. So, pharyngeal bars, there is a notochord, a dorsal hollow nerve cord, atriopore and atrium. These kind of go together, right? The oral hood is what brings the water into the pharynx. There are some what are called buccal cirri here, which are these little tentacles that act more like a sieve that prevent big things from being sucked into the oral hood and blocking it, right? And so they are very sensitive. And so they can have some sensation to them, some feel, some what they call mechanoreception to prevent big materials or from entering the oral hood. There are also some velar tentacles on the back here that also kind of act as a sieve and prevent too big of particles into entering the pharynx, so they can reject them and push them out the oral hood and back out into the environment if it's something large. These pigmented cells that are on top of those pigmented cells that are on top of the dorsal nerve cord are called ocellus cells, right? Just like the cells that we called them in neurochordates, and they can sense light so that the animal knows which way light is directionally. They're dorsally located. Now, if you look at a cross section of these guys through the oral hood, so if I were to section right here and pick the top or the most cranial part of the animal off and look in, right? This is what you see. So you got this big space, right? Which is the oral hood. You got a couple extensions here. Here's the opening to the oral hood. These are the buccal cirri. Here's the wheel organ, or part of the wheel organ on the sides that's heavily ciliated. And then at the top of, or what we call the roof of the oral groove, there are two smaller little depressions or grooves called Hack-Check's pits, or I call it Hack-Check's groove because it's a groove that runs back towards the pharynx. This is a structure that produces a number of hormones, one of them being growth hormone. And if you've had any human anatomy and physiology or any physiology before, you know growth hormone comes from the pituitary gland. So this is a primitive pituitary gland and is homologous to the pituitary gland in vertebrates. And it's named for the scientist who described it. That's where that name comes from, Hack-Check's groove. So this is a collecting chamber and also the pump, right? Part of the ciliary pump that pulls water into the pharynx. Now, this pharynx, which is slitted, has another name, and we've used the name once. We'll use it again when we get to lampreys. It's called the branchial basket. All right, branchial means basically, gill, pharynx, or a throat that does respiration. And it looks like a basket because of the bars and the slits when you look at it in 3 dimensions. We talked about the dorsal fin, so I think I've mentioned all of these. The intestine is broken down into a midgut and a hindgut. Midgut is obviously more cranially located. Hindgut is more dorsally located. Typically, what separates the two is a muscular ring called the ileocolic ring, which is just a thickened wall that's not always visible. But on our slides, I think you can see it on the slides downstairs. And this is kind of a valve that controls how much material can leave the midgut and enter the hindgut at any one time. Okay, so this next section is right about here. If we just cut right through the pharynx and pick the head off again and look in, then this is kind of what you're going to see in lab. So you can see I put this picture up here and showed you kind of where the dotted line is or where I'm sectioning. And so you can see, if you look in the middle of our cross section, there's the pharynx. And it's sitting in this white empty space, which is the atrium. When you look at the pharynx in cross section, you can start to see the structures we talked about. Here's a blow up of the endostyle, which is ventrally located or on the floor of the pharynx. And here's that other groove on top that's on the roof or the dorsal side of the pharynx. And this name has a name. It's called epibranchial groove, above branchia, the gills, or above the pharynx. And so the mucus is produced, and this endostyle runs the entire length from cranial to caudal, the entire length of the pharynx, as does the epibranchial groove. And the bars, the endostyle, and the groove are all lined with ciliated cells. Most of the mucus is produced by the endostyle and will be passed up, and it will coat all of those bars. So as the water passes through the bars, anything that's organic will stick to the mucus. The cilia of the pharynx beat upward and force the mucus up continuously so that it collects food and then is captured in the epibranchial groove. Then all the ciliated cells of the epibranchial groove beat caudally, right? They beat caudally towards the tail. And they will push all the mucus-laden food back to the esophagus and into the digestive tract. So this is a blow up right here of two of the bars in cross section. So here's one pharyngeal bar, here's another. You can see they're heavily ciliated. These ciliated are beating and helping move mucus and water through the slits between the bars. The other cool thing histologically is there's a blood vessel that runs through here, right? And the blood vessel that runs through the middle of each bar suggests that there could be some exchange of gases here also, right? Oxygen for carbon dioxide. As you move up or dorsally in this picture, notice once you get above the epibranchial groove, you got a really large circle. That's the notochord. And you're going to see some striations in the notochord. We're going to talk about the importance of those in a second. And then sitting dorsally to that or on top of the notochord is the dorsal hollow nerve cord. And then above that, most dorsally, is your little dorsal fin. Notice on the lateral aspects of the body of the organism are large muscle cells. They look a lot like skeletal muscle cells in cross-section that are called myomeres. So a myo means muscle, mere means unit. So they're divided into units, if you will, these myomeres. And they can contract to obviously bend the tail, right? And for locomotion. The other thing that you'll see a lot of times in these sections, particularly if you got a mature adult, is you'll see some gonad on either end here. This one's showing ovary. The big, huge cells are oocytes or eggs, developing eggs. Sometimes you'll see a testis, which looks different. It looks more like a tube with a bunch of thin strings or thread in it, and those are developing sperm. So you'll be able to tell the difference when you look in lab. These guys are dioecious, which means you have male and females, right? Remember dioecious, two houses, so you have separate sexes, male and female sexes. So far, so good? So a couple other pictures out there that I've grabbed. Some of these I took out of an actual textbook. This one I think I grabbed off of another university website that shows a cross-section through the pharynx. This is kind of more like what your images will look like tomorrow. All right, so this is a section, again, through the pharynx. Here are your two ovaries. So you see the big red cells. Those are each individual one is an egg cell. Nice pharynx, nice endostyle, nice epibranchial groove. And this drains into the atrium. You can see the slits. They're very numerous, right, in the pharynx. Here's a test. Here's a male over here. So female, male. So you can see here are some testes. This is a little further back, more caudally located, because now notice there's no pharynx, but you have the intestine. See that? So intestine there. Off the intestine is it's basically off the midgut. Off the midgut is a blind-ended pouch that sticks off laterally. It's called the midgut diverticulum, or the midgut cecum is another name that you'll see. And it's just a blind-ended patch that is homologous to the liver and pancreas in vertebrates. But this is what the testis looks like. So you can see it doesn't look like you have individual large circular red cells. You just have a tube. And this tube has got a very thin lumen or opening. And this really tall epithelium is where the sperm develop. If you were to zoom in, you could see developing sperm cells. So I could have picked a whole bunch of different things, but I just picked this picture here and another image of, I think, which is really interesting. One of my favorite structures in early chordates is what their kidney looks like. But this is a combination slide here that talks a little bit about, obviously it's labeled circulatory system, but I can also mention the digestive tract here. And so this is just if you could just rip out the pharynx with the intestine attached to it, right? And so you can see up here this would be attached to the oral hood, right? And we would be pushing water into the pharynx here. And then you have the slits, which are draining into, right? Draining into the atrium and then out the atriopore. And this shows you some of the blood vessels that you're actually going to be able to see, some of them. So for example, you'll be able to see some blood vessels in this area. You'll be able to see some blood vessels in the tail sections. And you won't have to know all of them, but there'll be a couple you have to ID in lab. But what is really interesting about this is particularly the way the blood vessels are set up around the pharynx is going to be very similar to how the blood vessels are set up in fish for gill organization. So again, this screams to an anatomist, at least, that the pharynx not only is used to collect food, but it's also used for respiration in this animal, right? Because the circulation is set up, at least, to be similar to the circulation that you see in fish for what we're going to call gill arches eventually that are incorporated in respiration. Now, interestingly, Amphioxus do not have a heart like vertebrates do. They have a more invertebrate-like setup with their circulatory system. It is a closed circulatory system. All right, so what's the difference between a closed and an open circulatory system? So we have a closed circulatory system. All vertebrates have closed circulatory systems, which means that the blood is contained in a series of blood vessels that are closed off to the rest of the body. That is, the blood doesn't bathe the organs like an invertebrate does. So we're circulating that blood, which means you probably have a little bit of blood pressure in there, right? Not a lot, nothing like what a heart can produce, but enough blood pressure to drive movement of the blood through circulation. Now, interestingly, if you start right here, there is a contractile sac that's called the sinus venosus. It's a word we're going to use a lot when we get to the circulatory system. So sinus means sac, right, or large cavity. Venosus means it's collecting blood from veins, all right, from venous circulation. So all the veins are returning blood back to the heart. There's no heart here, right? So the sinus venosus is going to be the chamber that collects all of the blood in vertebrates and delivers it to the heart. But notice there's no heart, right? But you can see there are veins, common cardinal vein, hepatic portal vein, right? They're draining into the sinus venosus, and then this can contract and force blood towards the head through a blood vessel that's running underneath or ventrally. This is the belly side of amphioxus. That's going to be called the ventral aorta, right? Now, it's running right through the middle of the endostyle. So it's also called the endostylar artery, which is homologous to the ventral aorta in vertebrates, right, or in fish. So you might wonder, why are we making that distinction? Because remember, the whole floor of the pharynx is endostyle, right? But in us, this is where the thyroid is. It's only one, it's a little small organ. It doesn't run the length of our throat. So it's not called the endostylar artery anymore. It's called the ventral aorta, if you have an aorta ventrally. Now fish do. Fish have a ventral aorta, just like amphioxus does. And what they're going to do is the blood that comes up is going to be sent dorsally through what are called branchial arteries, right? Branchial arteries. So again, branchial means what? It means gills, right? And so you can see these connection points that are sending blood dorsally, right, up to two blood vessels that are running along the length of the pharynx on the dorsal side. They go all the way up to the head. These are called dorsal aortae. When you see AE at the end of a word, it means plural. There are two of them, right? Again, common in fish to have two aorta or aortae, and they basically are collecting the blood to the head, and then they circulate it backwards. And notice the two dorsal aortae that are moving caudally eventually merge to form a single dorsal aorta that will go all the way back to the tail, delivering blood to the dorsal end of amphioxus. So they're passing blood up through the bars, right? Which means they're probably exchanging oxygen for carbon dioxide. So this is a lot like how blood vessels are set up in the gills, and these will be called gill arches, these branches that connect the ventral and dorsal aorta together. Now, what's really interesting about this particular organism, since I don't have a heart besides this little sac called the sinus venosus, each branchial artery here has a little swelling on it. That's called a bulbulus, which is a thickened epithelial region that's called a myoepithelial cell. And I know it's hard to see there, but that word there is myoepithelial cells. So there are myoepithelial cells that line each bulbulus, and there are myoepithelial cells that line the sinus venosus. These are contractile epithelial cells. They're not muscle cells like you find in the heart, but they are cells that can contract and relax, contract and relax. So you kind of have like multiple hearts, if you will, right? This is very similar to what you see in invertebrates, like worms. Segmented worms, for example, have multiple aortic hearts. And so that setup is very invertebrate-like. But each one of these can contract and force blood up into the dorsal aorta. So it's almost like you had a combination vertebrate-invertebrate circulatory system here. Kind of cool how it works. Now, if you go back here, as I mentioned, if you're food and you're collected by the epibranchial groove, you'll be passed into the esophagus, which will drain into the midgut, right? Coming off the midgut, or what they're calling the intestine here, is this blind-ended lateral sac that's called the midgut diverticulum, or the midgut cecum. And what's really interesting about it is the organization of the circulatory system is very similar to what you see in the vertebrate liver. The blood vessels going to the midgut cecum is called the hepatic portal vein, right, which is the same vein that delivers blood to the liver from the digestive tract. invertebrates. Recent studies have shown that the physiology of the midgut cecum is to do a lot of liver function, like store glycogen, detoxify, remove toxins, filter what's being digested, and to finish digestion. Also has an immune function that's very similar to some of the immune tissues that we have in vertebrates. But it is very liver-like and is considered to be homologous to what is eventually becomes the liver in the vertebrates. Okay, I think that's about all. So, any questions? Watch my time. Okay, my favorite structure. Guarantee this will be on the test. Always is. So this is one of the most unique structures in chordates. So this is kind of what we were calling the excretory system in Amphioxus. So in us, we have kidneys, right? Our kidneys, what do kidneys do? They filter blood. And they remove all the waste out of the blood and dump it into the urinary bladder, and then we get rid of it, right, to the exterior. These guys also have an excretory system that filters blood. And where you find this setup, right, this system, is in the pharynx, the dorsal side of the pharynx. So if you go up, and here's a cross-section through the pharynx. Here's the pharynx right here. Down here, eventually, we have an endostyle, and then up here, right here, we have the epibranchial groove, right? On either side of that groove, in the inner, kind of in the in-between tissues, between the groove and the notochord, so in this area right here, where the box is, there is a organ, all right? So when you look here, this is a blow-up of that area. It's this area right here under this box. Here's a blow-up. This is a drawing, but this is an actual blow-up from one of the slides. And what you're going to see is there's a blood vessel right here that sits on just lateral on either side to the epibranchial groove, which is this guy right here. And notice there is a tube, if you will, on either side. And then there's this structure here and this structure here. These are the excretory organs of Amphioxus. And this is a drawing of how this is organized. It's really hard to find really good images of this. I would really like to redo the histology of it someday. I've said that for 20 years. But I need to take a trip to Chesapeake Bay. That's where I need to go. That's where you can collect, I guess, lots of Amphioxus. But I've never made that trip. But if anybody would, if I could find somebody to send me live Amphioxus, I could probably redo this study. So these are the pictures that you see in the textbook, which are okay. But I'll try to describe kind of how this is set up. Now, what's really cool about this system, or really interesting, is it has both invertebrate and vertebrates kind of combinations of excretory units that you see, for example, in a flatworm versus a vertebrate. So it's like an intermediate that's got all mixed up, if you will. So it works like this. The reason that you have kind of invertebrate components to this is because there's no heart, right? The heart, or blood pressure, is what drives kidney filtration in us, right? In a vertebrate. We have a really strong heart. We have high blood pressure. High blood pressure allows for filtering to happen at the kidney. Well, we just did the circulatory system. There's no heart, right? So if there is blood pressure, it's really, really weak. So its filtration power is basically nil, right, for a kidney. So they have to rely on a different mechanism to draw the waste out of the blood. And they're going to use a specialized cell here that's called a selenocyte. All right, a selenocyte. In some textbooks, they'll label this cell a podocyte. All right, so a podocyte is the name that you would give it in a vertebrate. Podo means foot, site means cell, literally foot cell, right? Seleno means tube or tubular, cylinder, site, tubular cell. And this is what the cell looks like. It is basically a combination of a podocyte and a cell that you would see in Platyhelminthes that's called a flame cell. All right? It's the name given to it. It's called a flame cell in their excretory system. So flame cells are found, for example, in flatworms. And what a flame cell has is long microvilli with a cilium, or a really long cilium that looks like a flagella to us. And there are long flagella that come off of the cell body here of the selenocyte. So this whole thing is a selenocyte. All right, and you can see, if we just kind of look at it, its basic anatomy. Up here, this is the cell body. This is the nucleus, this circle. And you can see what they line up so that they are surrounding a group of capillaries that is called the glomerulus. All right? So this is a term that you see in a vertebrate kidney. So the word glomerulus, when you look it up, the Greek breakdown means ball of yarn. All right, a ball of yarn. So imagine taking yarn and balling it up into a fist or into a ball. And that's what it looks like under a microscope, a bunch of twisted ropes that are balled together. That's where the name glomerulus comes from. So if you notice here, check this out. Here's their dorsal aorta up here. There's a blood vessel that comes, there are two of them, right? Dorsal aorta, there's one here and one on the other side. They're going to send a blood vessel down that becomes a ball of capillaries called the glomerulus. And that ball of capillaries is going to be surrounded by these selenocytes. They're going to surround the ball of capillaries, forming a little sac, if you will, right? Or a little contained area. And they are going to drain into a nephridial tube, which is this tube right here. So notice they're basically packing this area surrounding this ball of capillaries, and then they send their long microvilli down into this tube. Notice the tube runs from where the selenocyte cell bodies are found down and empties into the atrium. Do you see that? And this is called the nephridial tube. Nephridia, the word nephridia means kidney. All right, so nephridial tube is this hollow tube that connects the glomerulus and the selenocytes to the atrium. Pretty weird, right? And unique anatomy. And so there are flagella that come off of really long microtubule structures, flagellum, just like our flagella, that come off of the cell body of these selenocytes, and they beat. And the microvilli are moving around. And this is exactly what a flame cell looks like in Platyhelminthes. And they call it a flame cell because it looks like a flickering flame under a microscope. And so this is the flame cell portion. And then the cell body also forms these little feet that are called pedicels, which form the wall of the sac that surrounds the glomerulus, all right, or the capillaries. This part here, the cell body and the pedicels, pedicel means small foot. This looks just like what you see in a vertebrate kidney. So the top of the selenocyte is vertebrate-like. The bottom of it is invertebrate-like. Really weird. Really cool for a microscopist. I still get geeked out by how interesting or how weird the anatomy is. So say you got 1,000 of these that are, or hundreds of them, however you want to think about it, that form these interlocking pedicels that surround the glomerulus. Blood is filtered down to there, right? And these are all beating. They're sending their cilia and flagella down into that nephridial tube. They're beating, they're sucking or drawing water or fluid from the blood between the pedicels into the nephridial tubule, which then drains into the atrium. right? So you got all of the pieces of the puzzle organized to produce an extra motor, if you will, a flagellar motor that drives filtration of the blood. The really long microvilli are thought to collect certain waste products from the blood, pass them through the cytoplasm of the cell body, and then drop them through the microvilli also into circulation. Now, has any of the physiology been done? Hell no, right? I mean, this is all hypotheses based on anatomy. But remember, form equals function. You know, what you see determines kind of probably how it works. The paper from the 70s that does this is just really poorly done. The microscopy sucks, and the EM pictures are atrocious, and it's honestly not very good. I wish I could redo it. Anyway, that's kind of how it's set up. It's pretty interesting. And so you got kind of this combination excretory unit that filters blood that is kind of vertebrate-like and kind of invertebrate-like. So, this is one of your essay examples that you could do. I like this example because it's really strange, but it makes you, if you understand how it works, describe the anatomy first and then give me the function, right? So I like to use it to get you thinking that way. Cephalochordates, we just have this slide really. So we did the kidney and one other really interesting derived structure that is really unique to Amphioxus is their actual notochord, what the notochord looks like on the inside. If you remember, I told you guys that these are really good diggers, right? They dig down into the sediment with their heads out. So when they come to a spot where they want to kind of hang out, they'll swim up with their heads up and then they'll drop to the bottom and then they'll use their tails as a shovel to, bury themselves in the sediment. The ability to do that is based on, or I should say the effectiveness of doing that is based on their notochord and some specializations to it that are considered highly derived for a notochord. So as you guys know, the notochord is a hydrostatic structure, right? It is wrapped in really tough, non-stretchable connective tissue. And then the inside has a bunch of cells that hold a lot of water, a lot of fluid. And if you produce pressure inside of this, it'll push on the outer sheath. This is the notochordal sheath on the outside, is the dense connective tissue that's really tough and not stretchable. If you can somehow produce a pressure in there that will push out on the wall of the sheath, that will produce hydrostatic pressure or water pressure. The more pressure you produce, the more hard, if you will, or the more rigor there is to the tail itself, that it becomes less bendable and more like a weapon or a shovel, if you will, to dig into the sediment. And the way that we produce this extra water pressure, if you will, is through a series of muscles that are specialized to the notochord. They're called notochordal muscle cells. And they are skeletal muscle. They look a lot like the myomeres as far as what their architecture or what their structure is. So if you were to look at a myomere, like you did on the slides, you would see that they're broken up into little units, right? And those units are like skeletal muscle if you were to look at them under an electron microscope. The notochordal muscles look a lot like them. They're thinner and in sheets that run in consecutive sheets that are spaced apart pretty evenly. They run pretty much the length of the notochord. And when they contract, they squeeze the water against the wall of the notochordal sheath and produce more pressure, making this notochord more stout and harder to bend so they can use it to dig into the sediment really nicely. If they just want to swim really fast, then they want their notochords to be really bendable. They relax these muscles so that the hydrostatic pressure is less and the rod or the notochord is more bendable. So they can contract and relax these muscle tissue within the notochord to kind of produce varying degrees of bendability and rigor to the notochord. And they use that when they're swimming. Now, what's interesting is we know the dorsal hollow nerve cord sits on top of the notochord or dorsal to the notochord. And the myomeres are on either side of it, right? Dorsally located. Oh boy, forgot to turn my phone off. That's my son too. Uh-oh, what does he want? Oh well. Damn kids. Sorry. Anyway, so the myomeres are lined up on top of or stacked on top of one another, and they have little nerves that run off of them that go into the dorsal hollow nerve cord. And the nerve cord is obviously part of the central nervous system and controls the contraction of those, right? Interestingly, the notochordal muscles also have these muscle tails, which are extensions of the muscle cell sheath that go up and through. They puncture the notochordal sheath and go up and innervate or meet with a motor neuron on the ventral surface of the notochord, so that the on the ventral surface of the nerve cord, so that the nerve cord can control both the myomeres and the notochordal muscle in times of contraction. So it's a really complex setup and a very nice derived system here that the notochord in Amphioxus has in order to dig themselves, like this picture shows, into the sediment by producing more hydrostatic pressure. So those are the two kind of unique anatomical features, the kidney and the notochord, kidney we did yesterday, of cephalochordates. The last slide here is just using some of the terms for the exam. Possession of a notochord is apomorphic in deuterostomes. When you compare deuterostomes to all other animals, right? It's an apomorphy. It separates them from protostomes, for example, right? Is a synapomorphy in chordates, that is, all chordates have it, and is a plesiomorphy in vertebrates, because it's going to be replaced by the vertebral column. So there's kind of how you use those terms or see those terms used.